Hey guys, welcome to another episode of the Human AI podcast. This is part 2 of liver and glucose homeostasis. If you haven't watched part 1, I would recommend you do it because we're going to continue from where we left off, which might be a bit of an arbitrary point. Well, if you're unfamiliar with how the podcast works, Grok and AI and I talk about these topics. I'm very unfamiliar with them and Grok explains them. Please cross-reference everything you hear and take everything with a pinch of salt. However, I do hope you find these things enjoyable and informative. Okay, Grok, we left off talking about the entry of glucose into the cells from the blood, and I asked you why polar molecules struggle to go through a lipid membrane. And basically, you told me polar molecules have a non-even charge, but I don't get why that's a reason to not be able to go across a membrane.
Polar molecules like glucose struggle to cross the lipid membrane because the membrane's hydrophobic core, made of non-polar fatty acid tails, repels their uneven charge distribution, which prefers water's polar environment. Their partial positive and negative regions can't mix with the oily, non-polar bilayer, so they need specific transporters like GLUT4 to pass through. This ensures controlled glucose entry into cells for energy or storage.
So, hydrophobic, what does that mean?
Hydrophobic means water-fearing, describing substances like the lipid bilayer's fatty acid tails that repel water and polar molecules like glucose due to their non-polar, uncharged nature. This causes polar molecules to be excluded from the membrane, requiring transporters like GLUT4 for entry.
But what does it mean to fear water? What exactly are you fearing? Are you fearing the uneven charge? Is this true for other things besides? Is this true in general for other substances or specifically for water?
Hydrophobic doesn't literally mean fearing water. It describes non-polar substances like the lipid bilayer's fatty acid tails that don't interact well with water's polar, unevenly charged molecules due to their own lack of charge. This repulsion isn't specific to water, but applies to any polar substance, as non-polar molecules prefer to cluster with other non-polar molecules, like oils, rather than mix with polar ones like alcohol or glucose. In cell membranes, this property blocks polar molecules like glucose from passing, requiring transporters, and it's a general principle for any polar, non-polar interaction. Want to explore other examples or focus on glucose transport?
So how come sometimes we say opposites attract and now we don't say that?
The phrase opposites attract applies to electrostatic charges, like positive and negative ions, but in cell membranes, it's about chemical compatibility. Polar molecules like glucose with uneven charge prefer water's polar environment, while the non-polar lipid bilayer repels them. This isn't about opposite charges, but about like attracting like. Non-polar lipids mix with non-polar substances, not polar ones. So, glucose needs transporters like GLUT4 to cross the hydrophobic membrane, despite any charge-based attraction rules. Want more on membrane chemistry or glucose transport?
Okay, so I have a non-polar membrane. When you say non-polar, also we say always this shit about, phospholipid membrane, and we have like the hydrophobic tails and the hydrophilic heads or whatever that shit is. I don't understand it. Can you explain it?
The cell membrane is a phospholipid bilayer with hydrophilic, water-loving heads made of phosphate groups facing outward toward water and hydrophobic, water-repelling tails made of non-polar fatty acids facing inward, forming a barrier. Non-polar means the tails lack charge, repelling polar molecules like glucose, which can't pass through without transporters like GLUT4. This structure keeps the membrane stable while controlling what enters or exits the cell.
So does the membrane itself have different charges? Is the hydrophilic part charged differently from the hydrophobic part?
Yeah, the phospholipid membrane has different charge properties. The hydrophilic heads, made of phosphate groups, are polar and often negatively charged, attracting water, while the hydrophobic tails, made of fatty acids, are nonpolar and uncharged, repelling water.
So
This
the water, the oxygen is negative, the hydrogens are positive. Can't the half of the molecule attract the outside and the other half attract the inside? Is not how it goes.
Water's oxygen is partially negative, and hydrogens are partially positive due to its polar nature. But this doesn't let it attract both the hydrophilic, polar heads and hydrophobic, nonpolar tails of the membrane. The hydrophilic heads, with their charged phosphate groups, interact well with water's polar nature, while the nonpolar tails repel water entirely, preventing it from passing through the lipid bilayer. Glucose, also polar, faces the same issue and needs transporters like GLUT4 to cross, as the membrane structure doesn't allow half and half attraction to bypass the hydrophobic core. Want to dive into how specific transporters work?
Okay. Let's talk a bit about the transport now, or with these channels. So we have this channel-mediated kind of transport. You mentioned GLUT4, and we have passive transport, we have active transport, down and against the concentration gradient. Talk to me a bit about channel-mediated versus carrier-mediated. We have both in the case of glucose or not?
Glucose transport into cells, like via GLUT4, is carrier-mediated, using specific transporter proteins that bind glucose and move it across the membrane, typically via facilitated diffusion, passive transport, down its concentration gradient, without energy input. Channel-mediated transport, which involves pores for ions or small molecules, isn't typically used for glucose, as it requires carrier proteins like GLUT transporters to bind and shuttle it across the lipid bilayer. Unlike active transport, which moves substances against a gradient using ATP, glucose transport via GLUT4 is passive, driven by concentration differences, but insulin regulates GLUT4's presence on the membrane. Want more on GLUT4's mechanism or other transport types?
I want us to get more into GLUT 1 to 5, but I want to end this conversation with hydrophobic stuff. And so we have why is the membrane non-polar? Because how come it's non-polar, but also it has a hydrophobic part and a hydrophilic part? So surely the fact that there's a hydrophobic and a hydrophilic part means that it's polar, no?
The cell membrane isn't entirely nonpolar. It's a phospholipid bilayer with hydrophilic polar heads that have charged phosphate groups and hydrophobic, nonpolar tails made of uncharged fatty acids. The membrane's core, formed by the nonpolar tails facing inward, creates a hydrophobic barrier that repels polar molecules like glucose, while the hydrophilic heads face outward, interacting with water. This dual nature doesn't make the whole membrane polar, but gives it distinct polar and nonpolar regions, with the nonpolar core being why glucose needs transporters like GLUT1-5.
So the polar heads interact with the polar water, but then they can't go through the hydrophobic tails. They just reach the heads.
Exactly. The polar heads of the phospholipid bilayer interact with polar water molecules outside the cell, but polar molecules like glucose can't pass through the hydrophobic tails in the membrane's core due to their nonpolar, water-repelling nature. This hydrophobic barrier blocks polar substances, forcing glucose to rely on transporters like GLUT1-5 to cross. Want to jump into those GLUT transporters now?
Well, okay, that makes sense. And has anyone done created any technology that allows water to go through the membrane without channels?
All right. You want to dive into glucose transporters, specifically GLUT1 to
No,
5?
I don't. I asked you something very specific. I asked, has anyone created a technology where the water can go through the membrane without using channel proteins and facilitated diffusion?
No technology currently exists to allow water to pass through a cell membrane without channel proteins, like aquaporins or facilitated diffusion, as the lipid bilayer's hydrophilic core inherently blocks water's polar molecules. Research into artificial membranes or synthetic channels mimics aquaporins but still relies on specialized structures to facilitate water movement, not entirely bypassing these mechanisms. Studies, like those on biomimetic membranes, aim to replicate this, but don't eliminate the need for some form of facilitated transport. Want more on these artificial systems or why water needs help crossing?
So the answer is no, basically.
Yeah, exactly. No tech fully bypasses channel proteins or facilitated diffusion for water to cross the cell membrane. The hydrophilic lipid bilayer still requires some kind of specialized structure, like artificial channels, to let water through. Want to pivot to those GLUT
So
transporters or
there's no
something?
way to like change the tails, like to become less hydrophobic, maybe.
No practical way exists to alter the lipid bilayer's hydrophilic tails to make them less hydrophilic without disrupting the cell membrane's structure and
I
function.
see. What about creating some distance between the two phosphate heads and tails without disrupting function and structure, just temporarily, so they can go back to where they were after?
Temporarily increasing the distance between phospholipid heads and tails to allow water passage without disrupting the membrane's function is not feasible with current technology, as the bilayer's stability relies on the tight packing of hydrophilic tails and polar heads. Altering this spacing, even briefly, risks destabilizing the membrane, affecting its barrier role and protein anchoring, with no known method to reverse it reliably without damage.
Okay. Okay, now let's talk a bit about glucose transport, GLUT1 to 5. So we said glucose enters by facilitated diffusion and it's a carrier mediated process. So what's the difference between carrier mediated and channel mediated?
Carrier-mediated transport, like with GLUT1 to 5, involves specific proteins binding glucose and changing shape to shuttle it across the membrane, relying on facilitated diffusion down its concentration gradient without ATP. Channel-mediated transport uses pore-like proteins, like aquaporins for water, to allow rapid passage of small molecules or ions, without binding, also typically passive. For glucose, GLUT1 to 5 are carriers, not channels, with GLUT1 in red blood cells and brain, GLUT2 in liver, GLUT3 in neurons, GLUT4 in muscle and fat, insulin regulated, and GLUT5 for fructose and intestines. Want more on specific GLUT roles or transport mechanics?
So glucose entry, it's so these GLUT transport proteins, we have GLUT. Let's start with GLUT1. So we have GLUT1. What's GLUT1?
GLUT1 is a glucose transporter protein that facilitates glucose diffusion across cell membranes, mainly in red blood cells, the brain, and the blood-brain barrier, moving glucose down its concentration gradient without energy input. It's crucial for supplying glucose to energy-demanding tissues like the brain, which relies heavily on glucose for ATP production. Mutations or issues with GLUT1 can cause neurological disorders like GLUT1 deficiency syndrome. Want to move to GLUT2 or dive deeper into GLUT1's role?
Let's talk about GLUT2, yeah, so we can find it in the liver and in pancreatic beta cells.
GLUT2 is a glucose transporter in the liver, pancreatic beta cells, and intestinal cells, facilitating passive glucose transport down its concentration gradient, with a high capacity suited for areas with fluctuating glucose levels. In the liver, it allows glucose uptake or release based on blood sugar levels, aiding homeostasis, while in beta cells, it senses glucose to trigger insulin release. Its low affinity but high capacity makes it ideal for handling large glucose loads post-meal. Want to move to GLUT3 or focus on GLUT2's role in insulin signaling?
So, tell me about GLUT3 then. So, it's found in the brain.
GLUT3 is a glucose transporter primarily found in neurons and other brain cells, with high affinity and capacity for glucose, ensuring efficient uptake even at low concentrations to meet the brain's high energy demands. It facilitates passive transport of glucose across cell membranes, supporting aerobic metabolism critical for brain function. Its presence in the blood-brain barrier and neurons makes it vital for maintaining cognitive function and energy supply. Want to dive into GLUT4 or something specific about GLUT3?
Tell me about GLUT4 now, which seems to be the main one we've talked about.
GLUT4 is a glucose transporter found in muscle and adipose tissue, uniquely insulin regulated, moving to the cell membrane when insulin binds to receptors, allowing glucose uptake for energy or storage via facilitated diffusion. Its high affinity ensures efficient glucose transport during high blood sugar, like after meals, supporting glycogen synthesis or lipogenesis. This insulin-dependent action makes GLUT4 critical for glucose homeostasis and a key player in conditions like diabetes. Want to dive into GLUT5 or how GLUT4 relates to insulin resistance?
Yes, let's do GLUT5.
GLUT5 is a fructose transporter primarily found in the small intestine, kidneys, and sperm, facilitating the passive diffusion of fructose across cell membranes down its concentration gradient. Unlike other glutes, it has a high specificity for fructose, not glucose, aiding dietary fructose absorption in the gut. Its role supports energy metabolism in specific tissues, but isn't directly tied to glucose homeostasis, like GLUT4. Want to compare all glutes or dive into something else?
Yes, let's compare them.
GLUT1 to 5 are glucose or fructose transporters with distinct roles. GLUT1 in red blood cells and brain has moderate affinity for glucose. GLUT2 in liver and pancreatic beta cells handles high glucose loads. GLUT3 in neurons has high affinity for efficient brain uptake. GLUT4 in muscle and fat is insulin regulated for glucose storage, and GLUT5 in intestines specializes in fructose transport. Each uses facilitated diffusion, but their tissue-specific locations and affinities, e.g. GLUT3's high affinity versus GLUT2's low, tailor them to local metabolic needs. GLUT4's insulin dependence sets it apart, while GLUT5's fructose focus makes it unique. Want to dive deeper into their regulation or a specific GLUT?
So, tell me again how GLUT4 connects with insulin. So we have an insulin receptor, insulin comes and binds, and then what goes on from there?
When insulin binds to its receptor on muscle or adipose cells, it activates the PI3K ACT signaling pathway, triggering vesicles containing GLUT4 to move to the cell membrane. Once on the membrane, GLUT4 facilitates passive glucose transport into the cell, driven by the concentration gradient for energy or storage. This insulin-dependent process is key for lowering blood glucose after meals. Want more on the signaling pathway or its role in diabetes?
Tell me about PI3 kinase, rapid translocation, and PKC, and how these play a role.
When insulin binds to its receptor, it activates PI3 kinase, PI3K, which generates signaling molecules that trigger the AKT pathway, prompting rapid translocation of GLUT4 vesicles to the cell membrane for glucose uptake. Protein kinase C, PKC, can also contribute by modulating insulin signaling, enhancing GLUT4 movement in muscle and fat cells. This coordinated action ensures efficient glucose transport to regulate blood sugar post-meal. Want more on how this fails in insulin resistance?
So let's talk a bit about the effects of insulin. So we can have immediate and long-term responses. So tell us about the differences. So we have the glucose uptake immediately. We have this modulation of enzyme activity. What would be some longer lasting effects?
Insulin's immediate effects include rapid glucose uptake via GLUT4 translocation to muscle and fat cell membranes and quick modulation of enzyme activity, like activating glycogen synthase for glucose storage. Long-term effects involve regulating gene expression, promoting protein synthesis, and enhancing cell growth or differentiation, such as upregulating lipogenic enzymes for fat storage over hours or days. These actions ensure short-term blood sugar control and sustained metabolic adaptations for energy balance. Want to dive deeper into specific enzymes or insulin resistance?
Tell me about how come we have an increase in expression of liver enzymes that synthesize glycogen? How is this a longer lasting effect of insulin? What about the increase in expression of adipocyte enzymes that synthesize triacylglycerols? Inhibiting lipolysis? How do these things occur?
Insulin promotes long-term increases in liver enzyme expression, like glycogen synthase, by activating transcription factors via the PI3K ACT pathway, enhancing glycogen synthesis over hours to store glucose. In adipocytes, insulin upregulates lipogenic enzyme expression, like fatty acid synthase, to synthesize triacylglycerols, while inhibiting lipolysis by suppressing hormone-sensitive lipase through similar signaling, promoting fat storage. These gene expression changes, unlike rapid GLUT4 translocation, take hours to days, ensuring sustained energy storage for glucose homeostasis. Want more on specific transcription factors or lipolysis inhibition?
Sure.
All right. You want to keep going with insulin's effects on enzyme expression or pivot to something else in liver and glucose homeostasis?
You just recommended something and I said yes.
Got it. My bad for the pauselets. Dive into transcription factors for insulin's long-term effects. Insulin activates transcription factors like SREBP1C and IREBP via the PI3K ACT pathway, which bind to DNA in liver and adipocyte cells to boost expression of enzymes like glycogen synthase for glycogen synthesis and fatty acid synthase for triacylglycerol production, while also repressing lipolysis genes. These changes, taking hours to days, ensure sustained glucose storage and fat accumulation for energy homeostasis.
Okay,
Want to fo-
let's talk a bit about the pentose phosphate pathway now.
The pentose phosphate pathway, PPP, in the liver cytoplasm converts glucose 6-phosphate into pentose sugars like ribose 5-phosphate for nucleotide synthesis and produces NADPH for fatty acid synthesis and antioxidant defense. It runs in 2 phases. The oxidative phase generates NADPH and ribulose 5-phosphate, while the non-oxidative phase converts these into other sugars for cellular needs. This pathway supports liver glucose homeostasis by balancing energy metabolism and biosynthetic demands. Want more on its regulation or role in liver function?
I don't understand what it is. Could you explain it very simply, please? And tell me how it branches from glycolysis at G6P. Tell me about ribose phosphates used to synthesize RNA and DNA as a product, and also NADPH. What's that? And we use it for biosynthesis and redox balance maintaining.
The pentose phosphate pathway, PPP, is a side path from glycolysis where glucose 6-phosphate, G6P, in the liver cytoplasm is used to make ribose 5-phosphate, a sugar for DNA and RNA synthesis, and NADPH, a molecule for building fats and fighting oxidative stress. Unlike glycolysis, which breaks down G6P for energy, the PPP branches off at G6P to produce NADPH in its oxidative phase and ribose 5-phosphate in both oxidative and non-oxidative phases for biosynthetic needs. NADPH is crucial for synthesizing fatty acids and maintaining redox balance by neutralizing harmful reactive oxygen species in the liver. Want more on how the PPP supports liver metabolism or specific products?
Tell me about these PPP enzymes, these PP intermediates, how they're recycled back into glycolysis. What's going on? And connect it to everything else we talked about, because I can't connect it. Just in 2 sentences.
The pentose phosphate pathway, PPP, uses enzymes like glucose 6-phosphate dehydrogenase to convert glucose 6-phosphate into ribose 5-phosphate and NADPH, with intermediates like xylulose 5-phosphate and glyceraldehyde 3-phosphate recycling back into glycolysis to support glucose metabolism and energy production in the liver. This connects to glucose homeostasis by providing NADPH for fatty acid synthesis, like insulin-driven lipogenesis, and ribose phosphate production, while integrating with glycolysis to balance energy and biosynthetic needs in tissues like the brain and muscles.
Okay, I want 1 sentence of tissue-specific glucose metabolism.
Tissue-specific glucose metabolism varies, with the liver regulating blood glucose via glycogen storage and gluconeogenesis, muscle and fat using insulin-driven GL4 for glucose uptake, and the brain relying on GLUT3 for constant glucose supply due to its high energy demand.
And 1 sentence of a key takeaway.
The liver orchestrates glucose homeostasis by using insulin and glucagon to regulate glucose storage, release, and metabolism, ensuring energy supply for tissues like the brain via pathways like glycolysis and the pentose phosphate pathway. Thanks for the episode. Peace out.